Abstract.
In this practice, the preparation of chemocompetent and electrocompetent Jacobi cells for the introduction of exogenous DNA was carried out. The transformation efficiency was measured for results obtained in Team 2 because for Team 1, no growth of transformed bacteria was reported. Similarly, for the different methodologies, it was possible to know the basis of the phenomenon of bacterial transformation and the differentiation between the chemocompetence and electroporation protocols.
Introduction.
The transfection is the one in the production of external genetic material in eukaryotic cells by means of plasmid viral vectors, in the case transduction is also spoken, or other tools for the transference. On the other hand, transformation refers to non-viral transfers of genetic material in bacteria and non-animal eukaryotic cells such as fungi, algae, or plants. Of the methods that are described in the practice, electroporation is the most suitable method for the transfection of eukaryotic cells. To know which method to use for the transformation of bacteria, the following should be followed. Especially prepared bacteria are mixed with DNA, probably of ligation, for example. A heat shock is applied to the bacteria, which cause them to collect the plasmid. The plasmids used in the cloning contain an antibiotic resistance gene. Therefore, all bacteria are placed on a plate with antibiotics to select those that collect plasmid. Bacteria without plasmid die. Each bacterium with plasmid gives rise to a colony or community of identical bacteria that contains the plasmid. Several colonies are analyzed to identify one with the appropriate plasmid by PCR or with restriction enzymes, for example. A colony containing the appropriate colonies are analyzed to identify one with the appropriate plasmid by PCR or with restriction enzymes, for example. A colony containing the appropriate plasmid is allowed to grow on a larger scale and is used to produce protein or plasmid. Analyze colonies. Other methods to transform bacteria can be plasmid transformation, cell competition, and cloning.
Methodology. Preparation of competent cells. First, take 0.3 ml of top 10 bacteria and incubate in Little Britannia. Then incubate at 37 degrees for 3 hours. Put the flask in ice for 10 minutes. Separate the culture in half to obtain component cells. Centrifugate at 6,000 repetitions per minute for 4 rates for 5 minutes. For chemicompetent, recover the pellet and resuspend in 10 ml of calcium chloride. Centrifugate and resuspend the pellet in 2 microliters of calcium chloride and glycerol. Then incubate 10 minutes in ice. For electrocompetent, recover the pellet and resuspend in 10 ml of glycerol. Incubate in ice for 10 minutes and centrifugate at 6,000 repetitions per minute for 4 rates for 5 minutes. Eliminate the rest and resuspend in 2 ml of glycerol. Prepare 200 microliters of the cuts and stock at -70 rates. Measurement of transformation efficiency. For chemicompetent, put in microtube 40 ml of salt and 2 ml of plasmid. Incubate in ice for 10 minutes. Put in thermoblock at 43.4 rates for 45 seconds. Then incubate in ice for 5 minutes. Add 1,400 of stock and then incubate at 33 degrees for 30 minutes with agitation. Finally, spatulate 100 microliters in clear volcanic plate. electrocompetent, put in a microtube 38 microliters of salt and 2 microliters of plasmid. Transfer to cold cuvette and then put the cuvette in ice. Apply electric shock and incubate in ice 5 minutes. Then add 1400 of soak. Incubate at 33 degrees for 30 minutes with agitation. Finally, spatulate 100 microliters in little Bertani plate.
¡Qué técnica!
Todavía no está completo su bagaje.
Results. After 24 hours of incubation, no colony-forming units were obtained in either of the two transformation methods, so efficiency could not be obtained since it would be 0.
Discussion.
The transformation of bacteria has a great relevance not only in the field of microbiology, but also as a storage technique and replication of DNA sequence or plasmids. Through chemical techniques or by electric shock, we can achieve efficient transformation of cells. As can be seen in the following figure, electrocompetent cells were prepared by electroporation, obtaining after spatulation that 8 colonies were formed with an efficiency of 1.184 times 10 to the third power colony forming units per microliter. Comparing it with efficiencies of commercial cells such as those offered by Thermo Fisher from magnitude 10 power 9, we can say that our efficiency in electrocompetent cells is low. This may be due to the fact that the sample was contaminated with salts from the juice buffer or that already came with excess of salts, and that is why many cells die, due to the electric shock to which they were subjected. This excess of salts causes the cells to conduct the electricity of the pulse that is given to them and then explode. It can also be seen in the figure that no colonies grew on the plate used for chemocompetent cells. As there were no colonies, the efficiency could not be calculated. This may be due to a critical point during the procedure, which is the thermal shock. Perhaps the incubation was not carried out at the correct temperature for the right time, since the equipment was damaged and the temperature display shows a different value than the thermometer. It could also be that when the spatula was not correctly cooled down and the cells were burned, so they did not grow. In contrast, Team 1 did not obtain any growth in its place, therefore its efficiency is 0. The problem maybe was in the spatulation, because the same samples, conditions, and regions were used in both teams. Conclusion.
The excellent execution of these protocols of electrocompetence and chemocompetence is of utmost importance because it is the initial step for other protocols of transformation, conjugation, and transduction.